Okay, so today we're just going to show you how we poured the plates. We made an agarose solution, which is really similar to the agar that you used in the Petri dishes for the genetic recombination lab. And then we just heated it up in the microwave till it's a liquid and dissolved it. And now we're going to pour it in one of these gel beds. So you can see why this might be called a bed. It's kind of looks like a bed, and we tape the ends. And so we need to grab one that has Mr. Can you put a comb in it or show them what the comb is?
So here's the comb. The other one was missing it, and it's this particular white piece, and they call it a comb because obviously it looks like teeth on a comb. Each one of those combs is going to be depressed in your agarose gel and create a well. We're not going to use all of the wells, but that's just what the comb is for this particular setup.
Okay, so we'll pour it in. And then try to visualize if we've gone.
Give me a little bit more.
Okay, that's probably good. And then just like in a cooking show, we'll fast forward. So now here's our finished product. You can tell it got a little bit cloudier, and we can just take the comb out, and you can see that there are little depressions right there. And that's where we will be putting our DNA into.
So now what we're going to do is we're going to describe how we're actually going to load the gels. And one of the things that you're going to get is a packet that has your DNA samples in it. And so what we want to make sure, if you notice at the very, very top how there's just a little bit of liquid sort of attaching itself to the top of the foil, but if you take it and just sort of flick it, most of that liquid is going to come down. You're still going to see a little bit of it there, but that should be okay. So now we're going to load our gels.
Okay, so one thing, this quick strip, it's kind of like a foil on top. So I would suggest either you maybe take a pair of scissors, something sharp, and just kind of poke through each of the A, B, C, D, and E. Okay, then you're going to be able to get your disposable pipette in. If you were doing this in a lab, you would use, you want to grab a micropipetter to show them, Mr. Ger? I don't know where we stuck that.
Yep, there's one right here.
Or like an actual, like the blue. In a real lab, you'd be using something like this, which is a very precise measuring tool. But this actually takes a little bit of time to kind of master. So we're just going to use these really quick and easy quick strips. So you've got a disposable pipette. You've got all your DNA samples here, and notice the DNA is all labeled A, B, C, D, E, F, G, and H are empty. And so you're going to kind of pay attention to where you put each sample. So look at our gel here that we made, and you're going to kind of, just like we read from left to right, you're going to fill A, B, C, D, and E, so the first 5 rows. So go ahead.
So one of the things I want you to notice too about the micropipetter is that these are the smaller versions of the ones that we've used in the past. If you were to squeeze the bulb while you're trying to withdraw some of that fluid, it actually might create too much of a suction. So we suggest that you squeeze this part, and you're only going to create a little bit of a suction when you squeeze it because you're going to make sure that you draw up the correct amount.
Okay, and the nice part about this, like I said, it's measured already, so like Mr. Ger said, squeeze kind of lower on the pipette, and then you're going to kind of go in, and you're just going to suck up all the liquid in the first column. And then I'm going to go over here, and remember this gel is really, really fragile. It's like a piece of Jell-O, so when I go to fill it, I can't just stab it all the way down. A little extra on top getting out is okay, but you want to make sure it's all in that first well.
And if you notice, you know, this is a delicate material. She did a really good job of just filling the well. So just do it very slowly, very delicately. And now, if we sort of tilt it a little bit on its side, you can see right there, it's somewhat translucent, that entire well has been filled.
So it's like a little mini cup that you want to fill.
Okay?
Okay. And so then you're going to do that with the rest of your samples. So the one thing you want to do between filling your samples is you just have one disposable beaker. So, I'm sorry, there it is. And we're just trying not to waste material. So we can use one the whole time, but we have different samples of DNA. We don't want to get them mixed up. So imagine you're at a crime scene. You don't want to mix up the DNA samples. So if you just kind of suck up some DI water and dump it in your waste container, suck up a little bit more and dump it. You know, you probably don't have to rinse more than a couple times. Okay, so now I'm going to go in and get my next sample. So I'm just, oh, thank you. It's right in front of me here. Okay, so I'm going to fill B. And again, the trick about holding lower on the pipette to get better control. So I'm going to suck the entire sample up, and then B will be right next to A. I just kind of gently fill it up, like so. And again, I'm careful not to push it all the way through, because if I do that, I know that it will puncture the bottom of the little cup. Okay, so now that all of our wells are filled, I'm going to show you kind of this perspective from the side because it's kind of cool. You can see that they're literally like little cups that are full of liquid. That last one looks
I punctured the bottom of the auger. That's a what not to do move.
That's okay, that can happen. And then a little extra just kind of running down the top is probably not a big deal because remember the DNA is negatively charged. It's going to be attracted towards the positive end anyway, and the gel is going to kind of slow it down. But now we kind of have an interesting technique for, we want to kind of close the top of those little cups. So we need to use a lid. So we actually melted some agar. It looks just like water now, kind of a thick water. And we're just going to suck some up in a pipette. And we're just going to run a ribbon down the top of our gels. And that, once that solidifies, that should prevent any extra DNA from leaking out. And then the nice part about that, it makes it much easier to load. So I'm just going to put some on top, and then we're going to wait maybe about 5 minutes, and then we're going to load our gel that we put on top to kind of close up those little cups has dried, and we're going to put this into the electrophoresis chamber. So if you kind of notice, I'm going to put the wells near the red part on top. And if I'm in this group, I'm going to remember that my gel is in the upper left-hand corner, because if you can picture this, there is 4 more places to put gels. One here, one here, one here, and one here. So when everyone has loaded their gels, then we'll put this little holding strip on, and then we're ready to put the top on. And if you notice, the top has electrodes of different colors. So there's red, and the reason I know that red is positive is because if I follow this red all the way over, it went into the positive input over here. So I'm going to turn this on, and I want this to be at about 125 volts. I know it's a little bit low right now, but this should take anywhere between 20 to 30 minutes for this DNA to migrate. And so if Miss Juger can kind of zero in a little bit, you'll see little bubbles rising. And so that tells us that we've put a charge into the water, and now our DNA sample should be starting to move.